In Porous Media Lab, we look at dynamic processes that are taking place in porous materials such as soils and rocks. One task that we perform here in Porous Media Lab is creation of microfluidics, which are transparent representations of these materials, and therefore we can look into porous materials and see how processes are taking place.
Using microfluidics, we can investigate the behavior of fluids through those porous materials at the micro to nanometer scale. We can look at very small volumes of fluids in the range of femtoliters. As orientation, a human red blood cell has a volume of about 90 femtoliters. So you can probably already guess that microfluidics are also used in medical research, for example, to study a human microvascular network or disease like cancer, or in synthetic material like polymers in diapers. In our lab, we prepare those microfluidics mainly for application in the field of geoscience by a so-called photolithographic process, where we coat photoresin on a small chip as a miniature representation of our porous material. We do this in a clean room in order to prevent any contamination of our microfluidics by small dust particles that are naturally present in air.
This enables exploring processes such as growth of bacteria and biomass in soils and rocks, as well as drying of soils or clogging process inside reservoirs and two-phase flow, for example. As we move into sustainable use of the subsurface, understanding such a process is essential because we want to understand the response of the subsurface to human activities. And microfluidics enable such a fundamental understanding of it, which is mostly missing when we use more traditional approaches like exploring soil columns or rock cores, because soil and rocks are opaque. We cannot directly look into them using traditional methods.